Our project demonstrated that diabetes causes misregulation of systemic iron homeostasis in the liver and the iron-dependent generation of reactive oxygen species with far-reaching systemic consequences. Iron overload in the body is prevented by the hepcidin-ferroportin regulatory system. Hepcidin is a small peptide hormone which is produced by the liver. Hepcidin binds to the iron exporter ferroportin and thus regulates the absorption of iron from the diet and the iron release from macrophages that recycle aging and senescent red blood cells. In the previous funding period, we showed that as a consequence of diabetes, too little hepcidin is produced in the liver. Therefore, too much iron is absorbed from the diet and released from macrophages, and the iron concentration in blood increases. In the blood, iron is bound by the transport protein transferrin, which can be taken up by almost all organs and cell types. Iron then accumulates in diverse organs and causes the generation of reactive metabolites. To understand why too little hepcidin is produced in diabetes type 2, we studied a disease model, the DB mouse. Similar to our observations in patients with diabetes, hepcidin expression is reduced in these mice and systemic iron parameters are elevated. Our analysis further showed that ACT phosphorylation in DB mice dominates over the iron-regulated BMP-SMAD signaling pathway, which results in the downregulation of hepcidin levels. We interpret this data in such a way that metabolic alterations in diabetes type 2 cause the misregulation of iron homeostasis in the liver. This means that alterations in iron sensing in the liver as a consequence of diabetes are a novel diabetic complication. A second important question we asked in the first funding period was whether elevated iron levels are causal for the generation of reactive metabolites and diabetic late complications. To test this hypothesis, we crossed mice with genetic iron overload with the DB mouse model. Our experiments clearly demonstrated that in this mouse model, it is not the dicarbonyls, but rather reactive oxygen species that induce post-translational modifications. It is interesting to note that elevated iron levels even cause a reduction of methylglyoxal levels and therefore seem to be protective. Our further analysis focused on the kidney. We observed that iron overload reduces the protective level of carnosine, a finding that will be further followed with our partners in the research network. In addition, we demonstrated that markers for morphological kidney damage are increased as a consequence of iron-generated oxidative stress. In addition, we were able to demonstrate that iron accumulation can cause much more severe atherosclerosis, a typical diabetic late complication. We showed that the atherosclerotic plaques that spontaneously develop in the ApoE mouse are more in numbers and larger in size in conditions of iron overload. These results are really impressive. In the second funding period of our project, we will pose the following questions. Which pathological alterations involved in diabetes type 2 cause alterations of iron metabolism, and how? We will investigate the consequences of iron overload and oxidative stress-induced post-translational modifications on diabetic late complications, and this time we will focus mainly on macrophages, nerve cells, and the liver. And finally, we will ask the question whether pharmacological iron depletion reverts the generation of reactive metabolites and late complications. To sum up, an adequate regulation of iron homeostasis is of critical importance to prevent late diabetic complications.